Namaste to all the students. Khushbu Vidya, myself JV'n Dr. Khushbu Verma, Faculty of Agriculture and Veterinary Science. Today we will discuss about cell cycle checkpoints, and this session is related to course Cell Biology of program B.Tech FBT, B.Tech MTech FBT and B.Sc FN&D third trimester. So what are basically cell cycle checkpoints or restriction points? So there are cell cycle control mechanisms in the eukaryotic cells. These checkpoints verify whether the process at each phase of cell cycle have been accurately completed before progression into the next phase. If there is any kind of error, so cell will be arrested in that stage, it will not go further. And there are three main checkpoints that control the cell cycle in eukaryotic cells. They are G1 checkpoint, which is G1 restriction points. Next one is G2 checkpoint, and third one is Metaphase checkpoint. So first of all, we will discuss about G1 checkpoint. So this checkpoint is presently at the end of the G1 phase and before S phase. This checkpoint helps in taking the decision of whether the cell should divide, delay division, or enter a resting phase, means G0 phase. If there are unfavorable conditions for the cell division, then this restriction point restricts the progression to the next phase by passing the cell to G0 phase for an extended period of the time. This restriction point is mainly controlled by the action of CK1P16, which is CDK inhibitor P16. The inhibited CDK not bind with cyclin D1, hence there is no cell progression. Active cyclin D CDK complexes phosphorylated retinoblastoma protein, which is PRB, in the nucleus. Unphosphorylated PRB acts as an inhibitor of the G1 by preventing E2F mediated transcription. Once PRB get phosphorylated, E2F activates the transcription of cyclin E and A, then which then interacts with the CDK2 to allow for G1S phase transition. This brings the cell to the end of the first checkpoint, which is unphosphorylated RB inhibits the E2F. Next one is G2 checkpoint. So this restriction point is located at the end of the G2 phase. This checks the number of factors which are essential for the cell division. If there is any kind of incompletion, so cell will be arrested at, over there. And maturation promoting factor or mitosis promoting factor or M phase promoting factor, which is MPF, is a protein composed of cyclin B and CDK1. This protein promotes the G2 phase into the entrance of M phase. MPF is activated at the end of G2 by the phosphatase CHK, which removes an inhibitory phosphate groups added earlier. The main functions of MPF in this restriction point are a trigger triggers the formation of mitotic spindle, promotes chromosome condensation, causes nuclear envelope breakdown. If there are any kind of a damage are noticed in the restriction point, in this restriction point, then the phosphatase not activate the MPF, resulting in the arrest of cell cycle in G2 phase till the repair of the damaged DNA. This prevents the transfer of defected DNA into the daughter cells. Next and last checkpoint is metaphase checkpoint. This occurs at metaphase. Anaphase promoting complex APC regulates this checkpoint. This is also called spindle checkpoint. This checks whether all the chromosomes are properly attached to the spindle or not. This also governs the alignment of the chromosome and integrity of the spindles. If there are any kind of a mistake, then it delays the cells in entering into anaphase from the metaphase. So this is the sum up of this session, and this session is powered by Digital Version 2.0, Jayoti Vidyapeeth Women's University. Hope you are satisfied with this session. If you have any query, then mention in the comment box. I will resolve it. Thank you.